what I would like to do today with you is to walk with you through a frontier in medicine and to share with you the excitement for how this frontier is enabling us to ask new questions about health and disease, to interrogate in new ways our bodies in space and time. It is a frontier that is enabled by a transforming technology, a real radical breakthrough called cell reprogramming. But it's about so much more than just technology, because it's about changing our gaze, the way we look at patients, the way we look at ourselves, and it's something that is going to affect our lives in our lifetime. So, changing about gaze, it's basically about going from leaving things out, from excluding things and putting them in brackets, to including things, to bringing them back in fact, to the fore, in the petri dish of a lab with very important clinical implications. And what do I mean by that? Well, let's go to Michel Foucault, who is an archaeologist of knowledge, that's how he defined himself, and who was looking at how medical doctors looked at disease and patients in the 19th century. But that gaze is still very much with us, except that it's a legacy under strain. So in his words, to the pure nosological essence, so to the essence of disease, the patient adds his predispositions, his age, his way of life, and a whole series of events that appear as accidents. He who describes a disease, so the medical doctor, the physician, must take care to distinguish the symptoms that necessarily accompany it and which are proper to it from those that are only accidental, such as those that depend on the temperament and the age of the patient. It's, of course, the language of that time. But the punchline comes here. Paradoxically, in relation to what he's suffering from, so the disease, the patient is only an external fact. The medical reading must take him into account only to place him in parentheses, right? So we are looking at patients, but since we are thinking about diseases, the patient in its individual configuration must be bracketed out. Quite a contrast with the view of precision medicine. This is a website from the initiative that Obama launched last year. Several similar initiatives have, meanwhile, flourished around the world. And the idea here is that the things that we had to bracket out are actually the very things that we want to focus on. And this is becoming possible to envision through the conflation of a number of technologies. First of all, on the one hand, the possibility to sequence very fast at an affordable price our genomes, so to actually read our DNA. On the other hand, the amazing capacity to compute large amounts of data is the so-called big data deluge that I'm sure you will have already heard of. But today, I'm going to focus about what is here in the middle, one of the enabling technologies that allows us to bridge the gap between genes and the environment, genes and clinical data. And that is cell reprogramming. And so I would like, first of all, to thank my team members, because it's important to realize that in science, when we change the game in a team. And to paraphrase Newton, we actually changed the game only on the shoulders of giants who have changed the game before us and in fact, who set for us the table on which we can start playing the game to start with. So this is my team in Milan. And giants of game changing, like Conrad Waddington, in the 50s gave us tools for thinking about development by making an analogy of the very first cells in the early embryo to a marble that is sitting up a hill. And it has various options in going downhill, and these options are the various fates that can happen to that cell. It could become a neuron, it could become a skin cell, it could become muscle. And of course, you see that it's downhill. So the analogy here is between a development and gravity, a process that once it unfolds, it is irreversible. It can only go downhill. To the extent that another giant of game-changing, Spemann, reminds us that we are standing and walking, like me now, with parts of our body which could have been used for thinking had they developed in another part of the embryo. So crucial moments at the top of the hill, very early on, cells are still very plastic, but those decisions will unfold all the way through. Now, the game has radically changed. It has radically changed thanks to decades of research which culminated in the towering achievement of Nobel Prize Shinya Yamanaka 10 years ago, who showed us that by tinkering with just very few genes, 4 genes out of the 25,000 that we have, it was possible to reprogram a fully specialized cell, like my skin cell, that only knows how to behave as a skin cell, all the way back to the top of the hill, to bring it to that stage so that now, in vitro, in that petri dish I was telling you about, it can actually be moved forward again. So we go uphill, and then we can go downhill again, but we can do it in vitro. What are the implications? Well, the reason why this is changing medicine is because this is actually enabling to make the body outside of the body. What has been the problem so far, the key rate-limiting step in medicine? Not having access to the cells that we most wanted to have access to, the disease cells of a patient. Why? Well, because quite obviously these cells are inside people's bodies. Think about brain, but in fact, for most of diseases, this was and remains a fundamental problem. Now, however, you can actually externalize tissues and cells of the patient. Because what you do is you reprogram skin cells. You have these induced pluripotent stem cells. They are induced because we induce them to do this transition. They are pluripotent because they are at the top of the hill, so they can do all the tissues of our bodies, but in vitro. And they are stem cells because they can keep doing it basically forever. Once we externalize, once we bring them outside, they are immortal. And so, in the dish, we can build avatars of our tissues, representations of our tissues in both health and disease that we can mine and probe for a number of medical questions. And of course, it could even be possible that some of these cells then can go back into the patient to regenerate parts of its ailing bodies. But today, we are focusing on what we can learn outside of the body. And so, what does the I in IPS really stand for? Of course, the original meaning is induce, because we were downhill and we induced them to go back up. But of course, you can't help notice how that I resonates with the so many small i's that label the devices that populate our digital social life. Starting, of course, with the internet, one of the words that, in fact, Steve Jobs mentioned in his speech in which all of these small i's were actually connected to the launch of the iMac, and the internet with the idea of the constant connectivity. And if you think about it, these avatars are actually all the time potentially connected to us, because once you reprogram from your skin these pluripotent stem cells, they are there, they can be banked, they can be stored in lab, and you can go back to them to ask medical questions because these cells have your genome, and these are the cell types that you want. And of course, information, because it is true that in a couple of days we could sequence the genome of all of us here today, but the truth is that we still understand very little of that information. And the only way we go about closing that gap of knowledge is actually having a system in which we can experiment on that level of information. And now we have the way to do it, because it is outside of the body. I don't think I need to tell you why it is inspirational. You just need to look at me and, to how I'm excited about these developments in my and several other labs. And of course, the individual, because this is the thrust of this frontier. Thinking about that patient going from being within a bracket and being left out to actually coming to the fore in a petri dish in a way that samples the specific configuration of each one of us for a range of medical questions throughout our lifetime. So the challenge, of course, is monumental, because we have clinical data, we have genetic data, and we need to bridge them in vitro through a number of molecular features that we have to see and discover in these avatars of ourselves. And so I'm just telling you very briefly towards the end what we are doing in the lab in which we study cancer and neurodevelopmental disorders, so intellectual disability and autism. And I'm going to tell you just about a pair of diseases, and just to make sure that we are on the same page. Almost all of our cells have 46 chromosomes. These contain about 25,000 genes. The two diseases I'm briefly telling you about are due to the fact that very few genes, about 26 from chromosome 7, are actually present either in one copy less or one copy more. So most of us have 2 copies of each gene, including these famous 26 genes. But children with Williams syndrome have one copy less. Instead, children who have one copy more, so who have 3 copies, end up having autism or, at any rate, a severe speech impairment. So these are 2 conditions that give us also an incredible glimpse into foundational traits of the human condition, like the acquisition and use of language and the acquisition of social interactions. Williams children are gregarious, hypersociable. The clinicians used to refer them as having a cocktail party personality. If you have one copy more of the same genes, so a very subtle change, you end up with autism. So this is just a more detailed view because it's not only about cognition, but it's also at the level of the facial features, that changing the dosage, the quantity, the amount of genes, or very few genes, ends up in having these symmetrically opposite phenotypes. And so what we did was to actually reprogram skin cells from these patients and their families and build these avatars. And then ask the questions. Once we have brought these cells back to the top of the hill, like the marble I showed you, what can we learn? What is the impact of having one copy less or one copy more of these apparently fundamental genes? Well, we discovered surprisingly that already very early on, already once we are at the top of the hill, we find major alterations caused by this imbalance in dosage. And that this alteration then is amplified as you go down the hill. So the problem is seeded very, very early on when you are at the top of the hill. And so this is then a summary of our project and these kind of projects which can really be applied to any kind of disease that has a genetic component. Here you have a disease that has a combination of features, and you can now resolve them. You can destructure them in vitro by building from that patient with that genome, with that disease, the tissue types that together then you can reassemble to study what this disease is really about. And in fact, I refer to this as the factory of avatars because this is, for example, an induced pluripotent stem cells that we reprogram from one of these patients in the lab, and we are able to push it in vitro towards the progenitors of the face, like here, or towards the neurons of the cortex. To the extent that now, this is very recent data from my lab, we and others are actually pushing a new frontier, which is making from these cells not just neurons, but 3-dimensional aggregates that mimic the development of the brain cortex in vivo. But it's happening in vitro. So we have models, avatars, which recapitulate the step in which we form a brain, but again, outside of the body. And so I would like to finish with Arcimboldo, a real game changer in art, where this amazing skill of capturing humanness, features of the human condition, not only of human anatomy, by assembling other aspects of life and sometimes even non-life in a way that at the end you actually reconstructed this model of humanness. And the technology I told you about today, which is really a foundational tool for precision medicine, is about capturing the humanness of a specific patient in a dish by resolving and deconstructing the disease and then bringing it once back on into a coherent whole. A very exciting time, which again will hopefully affect all of us because it will give us new tools to push ahead this frontier. Thank you very much.